today, we're absolutely thrilled to welcome Tom Miller, CEO and co-founder of Iambic Therapeutics, to BIOS Builders. Tom, thank you once again for joining us.
It's a pleasure. Thanks, Chris.
Let's kick things off. Tom, would you mind starting us by sharing a brief personal introduction?
Sure. I come from Texas. I grew up in College Station, Texas, and went to school there. I went to Texas A&M as an undergraduate in chemistry and math, and kind of fell in love with the interface of those two subjects. After getting a Marshall Scholarship, I went to the UK for my graduate work, first at the University of London, then getting a PhD at Oxford. I came back to the States and did a postdoc at Berkeley before beginning a faculty career at California Institute of Technology, Caltech. And that was just a fantastic chapter of my career. I spent over 10 years as a tenure faculty member and building a research group there, having amazing students that have gone on to do extraordinary things, and really seeing the impact of the technology that we could create in the laboratory. And that kind of created the seeds for my own journey from the university and to build Iambic.
And we're about to dive a little bit deeper into that today. And thank you for that background. But before we do dive deep, any chance you could just provide a brief overview and introduction to Iambic for us, please?
Yeah. Iambic Therapeutics is a company that focuses on designing and discovering new molecules to address urgent unmet patient need. And we're really differentiated in three key ways in the way that we do that. We focus on developing leading proprietary AI technologies so that we can make more accurate predictions in the design of those molecules with fewer data points, enabling us to discover superior development candidates. We tightly couple those AI designs with high throughput experimentation and a platform that allows us to go from new molecular designs to new experimental data in a week. This allows us to execute closed loop design cycles that are necessary for our fast discovery process. And thirdly, we're really differentiated in the way in which we've used that platform to deliver new molecules to clinic at a pace of program launch to IND submission in 2 years. And that has allowed us to really demonstrate the value and the capabilities of the platform, and to expand upon that in the next chapter of the company.
And talk about some incredible speed. So let's start to dive a little bit deeper and maybe even take a step back to talk about the origin story. So in 2020, Iambic was founded out of your lab at Caltech with an incredibly bold mission of using physics-informed AI to really revolutionize drug discovery by illuminating that novel chemical matter to unlock better development candidates, and then using that high throughput discovery platform you talked about to more rapidly reach clinic. And if you're hitting IND in 2 years, just wow. But maybe taking a step back, any chance you could tell us a little bit more about the founding of Iambic? So what sort of prompted you to transition from academic to entrepreneur, and a little bit more about the spark that started it all in there?
Yeah, I would begin by saying that I really did, Caltech's an amazing place to work, and I absolutely loved working there. It's a place where the students are fantastic. The faculty members are an amazing group of people that are really pursuing just incredibly exciting journeys. And what we began to see during my later years at Caltech was the fact that the tools that we were developing were not only exciting as scientific capabilities, they were exciting because of their translational opportunity. They could have real world impact in terms of delivering better molecules, discovering new materials, and I wanted to pursue that as a way to see and to realize that translational impact. So Caltech was very supportive with that process. They kindly gave me a 2-year sabbatical, where, along with my co-founder, Fred Manby, we created a company that was initially called Entos and has since changed name to Iambic. And that company utilized those AI tools and demonstrated that they could actually be used to deliver better molecules in the space of drug discovery. And what has been an incredibly exciting journey is that on the basis of that demonstration, we've been able to develop an internal pipeline where we can really execute on the advancement of those AI-discovered molecules to clinic, and to build a team around that potential.
And only just because you've brought it up now, would love to ask a little bit about the name change. So Entos to Iambic, tell us more.
Yeah, sure. Iambic Therapeutics, first and foremost, is basically a recognition that with the completion of our Series B, we're embarking on a really exciting transition from being a preclinical company to a clinical stage biopharma. And to mark that transition, we wanted to adopt a name that still reflected our AI roots, but which nonetheless explicitly pointed out that we are a clinical stage therapeutics company. And I would say on top of that, Iambic as a name does reflect those AI roots because of the central role of large language models in the latest AI technologies that we're developing, and the connection to that is through the word Iambic.
No, I love that. And it really does seem to embody the story as well as that transition and emphasis on this next stage in Iambic's journey. But before we chat more about the approach, we've talked about this a little bit already, and you've been giving us some context. Can you tell us a little bit more about the difficulties surrounding traditional small molecule drug discovery? What's really been holding us back? How do better AI methods come into play? And maybe what have been some of the challenges around existing AI methods so far?
Certainly. I think that one of the things that makes small molecule drug discovery so challenging is just the fact that the space of potential small molecules is just extraordinarily vast. It's a combinatorially large space of potential molecules that could meet the needs of any particular drug program. And designing molecules that can satisfy the many properties that are needed for a successful clinical candidate is really the challenge of executing on that massive search. And the other thing that makes that hard, by the way, is the fact that even very small changes in the molecules, changes of one atom or changes of one chemical group, can have dramatic impacts that either make or break a potential drug. So what we have recognized and what we are embrace as a company is the fact that AI can really help in many ways in that search. On the one hand, it can help us for a particular target to identify the best way in which to effectively drug that target. Is a covalent molecule the best strategy? Is a non-covalent molecule the best strategy? Should we go for an allosteric mechanism or a protein-protein interaction? And having tools that can move between those different mechanisms of action is a very powerful way to address a given target and to explore the opportunities around that. Additionally, AI, and again, this is one of these three-part answers, but additionally, the AI is very effective for not only figuring out a good way to hit a particular target, but a good way to design an overall target product profile. It's not simply a matter of getting efficacy on a particular target. You want to have selectivity versus other off targets, or perhaps you want to have activity on two targets in a dual inhibitor mechanism. And being able to design molecules that can address those selectivity challenges is another tool that AI is incredibly helpful for. And then the third way in which AI is just incredibly powerful is to help us really navigate that large space, to find those islands of molecules that have differentiated property and that have the potential to really be effective in clinic.
So taking maybe a summary of that, and correct me if I err, traditional therapeutics discovery, it sounds highly complex. There are obviously prolonged timelines. And it often requires vast amounts of data where, given this complexity, where often I suspect that data is pretty scarce. So in Iambic's platform, you combine your proprietary AI methods with the high throughput experimentation to allow for more rapid design make test cycles, to explore those novel mechanisms of action, improve those TPPs, and ultimately produce better drugs. And this all falls under the OrbNet approach, which is a physics-based AI model, if I'm not mistaken. So can you tell us a little bit more about it?
Absolutely. So OrbNet is one of the key AI tools that we employ in designing molecules. And we've really developed a suite of such tools that address the various challenges of identifying molecules and leading development candidates. But OrbNet really was the first one that we developed, and we've published a fair bit about that. And really what that allows us to do is to use AI to more accurately solve the Schrodinger equation in the case of OrbNet. And that's basically the central operation for any kind of computational chemistry. It allows us to accurately make predictions about the energies in the binding of a particular molecule in its interactions with different protein environments. And that allows us to do that with an accuracy and a speed that was previously impractical. And AI was central to being able to execute that task.
And even just last year, I think you announced that the latest advances in OrbNet use geometric deep learning to more accurately solve the Schrodinger equation, just as you said, and that was completed in conjunction with CalNet and NVIDIA, if I'm not mistaken.
Yeah, it was a collaboration with Caltech and NVIDIA. That's right, yeah.
Could you tell us maybe a little bit more about that and how it got started, where it is today?
Sure. We've benefited from extensive collaborations with Caltech, of course, where I was formerly a faculty member, and with NVIDIA, who's played a very collaborative role in the development of a number of our technologies. And OrbNet is a great example of that. It takes a significant amount of engineering and novelty to really deploy the training of a platform of that kind. We train across very large numbers of physics-based calculations to accurately train the models, and we deploy that at a scale that allows us to really use that day in and day out for all of our drug discovery activities. And NVIDIA was a great partner in that process. And I would say that was really only the first chapter of the NVIDIA relationship. Even this last year, we've announced an entirely new technology called NeuralPlexer, which is an exciting tool that allows us to execute de novo protein structure prediction, not only for the isolated protein, but also in complex with another small molecule, allowing us to reveal cryptic pockets, to predict allostery, and to identify protein-protein interactions in a way that is an advance in terms of what has been possible in terms of the accuracy of those predictions. So this is... Yeah, so this is an ongoing relationship, and again, you'll see in the Series B press that has recently come out that relationship with NVIDIA remains alive and well.
Always exciting to hear, and you're right, that sort of collaboration can be a huge accelerator. But you mentioned the NeuralPlexer as well, and so maybe without going into too much detail, can you share more how Iambic systematically has incorporated and continues to incorporate those physical principles into its AI architectures? What's sort of the secret sauce behind the platform?
Yeah, I think that's a great question, and it is a unifying theme the way we try to tackle machine learning problems. I think the core recognition that we have is that physics and data are not in competition somehow. It's not a first principles computational chemistry approach that should be in competition with a purely data-driven approach. The reality is, by making AI architectures, by making machine learning methodologies that naturally incorporate as many of the physical principles as possible, lighten the load in terms of the data requirements. If you're not requiring the data to teach you things you should have known from the physics, then you can use that data for much less trivial tasks of making more accurate predictions. And that's reflected in the achievements of what the OrbNet architecture delivers. It's reflected in the way in which we design and execute NeuralPlexer, and it's a core aspect of the way in which we design AI in general.
I know this is a bit maybe broader scope, but it feels to me like in recent years, and maybe actually we should take a step back. A few centuries ago, it was incredibly common for other sciences, chemistry, biology, to really be informed in advance by those who are coming from physics. And it feels today, even in the biotech space, we're starting to see that again. The incorporations of microfluidics to make technologies more robust, high throughput, the incorporation of cryo-EM to inform data sets, or these physics-based principles applying to AI, really informing and accelerating drug discovery. It almost feels like physics is back at the fore, even in this space of biology. And of course, the wet lab always has to take front and center, but there are ways we can improve on this robustness of biology. And as someone who's made that transition, I mean, I would just, casual observation for me, just even during this call that came to mind, would love your thoughts.
Yeah, I think that's a great point. And I would say that, you know, as a newcomer to biotech, and I am a relative newcomer to biotech, you know, it's a field that is so hard in so many different ways. To have a successful drug really developed means that you have to surmount many, many different sorts of challenges simultaneously. And that really makes it a very natural playground for innovation of all kinds. And much of that innovation involves the ability to combine technology in that design pursuit. And anytime you're dealing with new technology, anytime you're dealing with new physical capabilities, at its heart is going to be a physics origin. And I think that's a natural source for the observation you're making.
And it means there's going to be a lot of fun ahead. So I'm excited for the field, for patients, and for really everyone involved. But Tom, thanks for helping lead that transition and drive us forward.
Absolutely. Thank you, Chris.
And from that, you've led to a number of drug compounds designed, which you've talked about. And now we're transitioning to that next stage of Iambic's approach. Your high throughput discovery platform, high throughput chemistry, and high throughput biology, forgive me, to execute those design make test cycles within a week, which is really just wild. So can you tell us a little bit more about Iambic's approach specifically to the high throughput chemical synthesis and how you're able to accelerate that initial development?
Certainly. So Entos has a fantastic medicinal chemistry team, not only utilizing conventional medicinal chemistry approach, as well as robotic synthesis and plate-based synthesis technologies. And we do those plate-based syntheses in conditions that are enable air-sensitive reactions, that enable flexible catalyst applications, and that effectively in a plate-based format allow us to do 1- and 2-step reactions that span 70 to 80% of the typical chemical reactions that would be in the repertoire of a medicinal chemistry team. And from a design and execution perspective, there's really a couple things that one has to work hard when one is dealing with that challenge. On the first hand, when you are designing the molecules to execute in any one of those syntheses, they have to be aware of the synthetic accessibility. It's no good to use AI to design molecules that are effectively impossible to make, or that would take a long time or impossible resources to actually make. So being aware of the synthetic routes and ensuring that you get expanse across chemical space while retaining that synthetic compatibility with the platform is a key challenge that we've worked to address. The other thing that one really has to work to address more on the experimental side, although that too dovetails with the AI, is the fact that robotic synthesis can be executed in 96, 384, or 1536 well plate format. And those 1536 well plate formats are really down at nanoliter volumes. They're extremely small volumes. You, of course, have the usual issues then of liquid handling at that small scale, which is essentially a solved problem. But nonetheless, you have to do quality control on determining what the reaction was. You have to do yield evaluation on those reactions. And you have to do all of that in the context of volume amounts that effectively preclude the ability to do purification. So you have to be able to assess all of those things using different mass spec technologies, dovetailed with AI technologies that allow you to answer those questions. And that's something that the team has worked tremendously hard on and has made great progress.
And it sounds like that comes together as you're talking about with QA/QC. The next step is always, of course, the biological assays that characterize those synthesized compounds and their impacts. And so as you're thinking about this sort of intersection with AI, with the physics-based principles incorporated, and how synthesis is done, how it's optimized, but also how the molecules are designed, how does that then integrate into the biology?
Yeah, it's been done in a way that seeks to make that as seamless as possible at Iambic. And really that involves developing both biochemical and cellular assays that can also be executed in a way that either enables purification or that can be executed in the absence of purification. And one of the tools that we do in that regard is affinity selection mass spectrometry. That allows for basically crude reaction mixtures to be subjected to the protein that would be the binder of interest. And then one can titrate in that protein concentration to determine the relative strength of binding of the different molecules that were synthesized. And that basically allows us in a purification-free way to execute biochemical assays. And we have similar tricks that we've developed for cellular assays.
No, it's phenomenal. And I mean, I assume as you're going through this at every point, the data being generated are feeding back into Iambic's AI platform, which is then informing how to improve the methods as you're moving forward.
That's incredibly important, to be able to not only execute the experiments as quickly as possible, but then to feed that back into the machine learning models so that we can fine tune and update the models on a weekly basis in a fully automated manner. And if you're making, 1536 well plates reactions, that's, a lot of titration curves every week that you're not going to be wanting to quality control as well from an individual. So having a fully integrated way to do the automated quality control, the automatic data processing on the basis of that leads into the retraining of the model is a critical part of the overall cycle as well.
I know we're really jumping ahead here with this question, but do you see this sort of shifting to become the standard for small molecule drug discovery and design in the future?
I think that elements of this are widely embraced. I think that Entos has really focused on, not only the differentiated AI tools, but really ensuring that the integration between that AI and the experimental execution is tightened as much as possible so that we can really execute as quickly in design make test cycles for the internal programs that we pursue as much as possible. I do think that will be an ongoing trend, and I think the sort of demonstrations that we've seen with our lead program provide a good demonstration of why that would be a fruitful direction to pursue.
And speaking of some of those programs, so Iambic is currently progressing a portfolio of 4 programs, initially focused on identifying drug molecules that could deactivate proteins linked to certain forms of cancer. Yeah. Would love to better understand what led you to make some of your initial focus criteria and what led you to select certain indications for your internal pipeline.
Absolutely. I think first and foremost, we focused on oncology as a space of urgent unmet need. There were targets that were clinically validated, that were clearly the right targets to be going after, and yet having a sufficiently effective molecule or a molecule with the appropriate profile to achieve efficacy and tolerability had remained elusive. So that had really guided our choice, providing, again, clinical validation, providing the opportunity for speed to market and speed to clinic, and the ability to really demonstrate that by making the superior molecule or making that first-in-class profile enables us to really demonstrate what we can execute on the platform quickly.
And speaking of that execution, Iambic's lead asset is a tyrosine kinase inhibitor that selectively targets HER2 and HER2 mutants. And you're actually scheduled to commence patient dosing in early 2024, which is truly an incredible feat, from program launch and frankly company build to IND submission in 24 months. So let's just pause and say, wow, you and the team have done something incredible here, Tom.
Yeah, the team. We should thank the team for that.
Love to chat more about them in a moment. But as we think about the applications, as we think about the speed, do you find that the technology is limited to any specific therapeutic areas, mechanisms of action, or is it really just a matter of point and shoot and figuring out where to apply it next?
The technology, I would say up front, is very agnostic to those different considerations. Even within our own pipeline, we try to utilize that breadth and flexibility. Our first program, as you mentioned, is a brain-penetrant HER2 selective inhibitor, and that it employs a covalent mechanism of action. Our second program is a selective CDK2/4 inhibitor, and that employs a non-covalent dual inhibitor mechanism of action. But our third program is actually an allosteric inhibitor, utilizing a cryptic pocket that we've discovered, and our fourth program is a PPI inhibitor. So it's actually utilizes that breadth. And although we've focused internally on oncology, I would say that the platform is again agnostic with respect to therapeutic area as it is to protein classes, and that's enabled us to engage in that flexibility through partnerships.
And that's actually where I would love to jump. So thinking about these, the incredible speed and the fast turnaround time from program launch to IND in 24 months that Iambic has been able to demonstrate with its now lead programs, and that we have to assume is repeatable for others, especially across different therapeutic areas. We'd really love to hear more as you make this transition from R&D company now into clinical stage biopharma company. This is a huge milestone, and we just love to learn more about your thoughts during this time.
Well, I appreciate that question, and I agree. It's an incredibly exciting transition for the company. And I think it's a great achievement for what the team has been able to execute on in such a quick pace. And I would really say that it's not just about the speed. As we embark on clinical entry with our lead molecules, the fact that we got here quickly is great and gives us a competitive advantage. But the reality is now it's the quality of the molecule, the degree to which it's actually going to provide that efficacy, provide that better therapeutic window. And really, the end of the day, the thing that matters most in any of these design processes is that resulting quality of the development candidate or clinical candidate. And I think that's the other thing that's exciting about the platform is the ability to deliver both those best-in-class and first-in-class molecules. And in terms of that transition, as a company, we're lucky to have deep clinical experience with our senior team, and of course we're building actively as we embark on that new chapter.
And we're going to be excited to see some of the results that are hopefully coming out soon. And as you just mentioned earlier, Iambic has not only seen incredible success since its founding in early April 2020, but you're recently announcing a $100+ million Series B, co-led by Abingworth and Ascenta. Maybe this is a good time to first not only say huge congratulations on everything you've accomplished so far, but maybe taking a step back, in your own words, what have been some of your greatest challenges on the way so far?
Well, it is an exciting chapter. The Series B is going to give us those years of runway, not only to take our molecules into clinic, but to be able to demonstrate for both of our two lead programs clinical efficacy. And I think that it's going to see the company really enter into a different phase of its journey. And what have the challenges been along the way? I think that when one goes from a company that was originally envisioned as a software company to a clinical stage biopharma in 2.5 to 3 years, you're dealing with a lot of change management throughout that process. People who initially joined thinking that they're going to be delivering on SaaS, suddenly get focused on executing on an internal pipeline to really trade on their own account, if you like, in terms of the execution of that platform, and then not only go through that discovery stage, but then to embark on the next stage of a clinical journey, has led to the need to make sure people are aligned, make sure that we kind of celebrate the interdisciplinary nature of our teams and our capabilities, and make sure that they remain fully understanding and engaged in where this company is able to go together.
Oh, and it seems like it's going to be going to some pretty great places. But as we think about some of the challenges, there are also obviously a number of phenomenal accomplishments. Are there any that have really, I know we've talked about the team. Are there any that have really stood out to you?
Yes, I would say the team. Again, I just can't say that enough. To have assembled La Jolla is an amazing place to build a biotech. To have recruited the experimental team, to have recruited the AI team, to have recruited the platform experts that we have as a company. I think that's that's an incredibly important accomplishment in general, and something that I'm so thankful of. And I would really say that the other thing that I would really find is incredibly validating from this journey is the fact that, in a sense, the thesis of the company has been demonstrated to work. To really, we entered into this company with the idea that if we could build these AI tools, if we could integrate them into a platform, we should be able to make better molecules. We should be able to deliver across different targets in a way that is faster and more effective, and leads to hopefully better therapeutics. And we're really seeing the realization of that idea, and the demonstration that we are able to get to clinic at a pace that is years faster than the expectations of the field, and really we're going to be able to enter that next chapter where we can demonstrate that they make a difference in the lives of real patients, and that's just an incredible thing to have the opportunity to participate in.
And maybe taking this a slight step back, refocusing on the team just for a moment, but loving that message. As we think about TechBio, this integration of technology into bioengineering platforms, it feels like we're seeing ever more increasing requirements for interdisciplinary teams. And as someone who's built such an incredible team over at Iambic, and to us now Iambic, how do you think about that interdisciplinary team and culture? Obviously, San Diego is a great place for it, generally speaking, especially with all of the folks from Illumina who've built out all the sequencers and everything there. But would, I guess, love to just hear your thoughts and maybe advice for people building similar companies.
I think that it's very important that there is a collective understanding of what we are trying to build together. I think that people need to see in a large interdisciplinary effort, not only what their local goals are, but how those local goals dovetail with the goals of the people around them. So it's not a collection of disparate parts, but really something that's building towards a single or a small number of unified goals. I think that's an important thing, and that takes a lot of work and a lot of alignment and a lot of transparency to try to make that happen. I think the other thing that we were fortunate to kind of appreciate early on is that by the very nature of interdisciplinary work, you are doing something that no one person can do. Nobody's going to be the expert at everything. You will have your, domain of strength, and that will, hand off to other members of the team. And to build a culture where you have that expectation of not everybody, or having no one person know everything, is a very important way to approach the problems. And I think building a team that had that collaborative approach, that had that understanding of the need for respect for other expertise has been a really critical part of building the fabric of the company.
I love the way you describe that respect for others' expertise and the combination being greater than the sum of its parts, 1 + 1 = 4, as it were. So having discussed Iambic and your approach to date, would love to try and ask and project out for the future. What's coming next for Iambic?
Well, I think we're getting the chance to continue to execute on the thesis of this company, and I think that is an incredibly exciting thing. We've got a technology roadmap for our continued advances on the AI front. We have active targets that are moving into clinic and that will be first in human as early as February of the upcoming year, and we'll be seeing that demonstration of the efficacy of what comes off the platform. We have the growth of additional programs and the expansion of our internal partnership, as well as the partnership of Entos with other companies and collaborations to take advantage of the bandwidth and the discovery capability of the platform across therapeutic areas. So I think that there's a lot that we can look forward to in the coming years at Iambic.
And there's a lot that hopefully we can have you back on to discuss some of those additional milestones of success.
Absolutely.
But maybe continuing to think out and project into the future, especially around that patient impact space and around these accelerated timelines of using tools such as OrbNet and the Neuroplexer. Thinking more broadly, are there additional technologies enabling this sort of AI for drug development that, you're building and/or would be excited to see emerge as one of, I would say, the leaders in the space, bringing them forward today?
I think there's no doubt that it's an exciting time for AI with the advent of large language models and transformer models, the wide utilization of those, and the integration of those with the discovery and content creation process. I think is an incredibly exciting place to be. I think that one of the frontiers that really makes or breaks this enterprise of using AI in drug discovery is to continue to innovate at the interface between the AI design and the experimental execution. I think that is a space where the boots on the ground efficacy of the approaches really come to matter, and I look forward to the continued innovation on that front as well.
I think we all do. And this has been a lot of fun, Tom. Before we come to a close, I did want to ask maybe a few rapid fire questions just to cap things off. So where are small molecules today? What challenges have we addressed? Diseases treated? Where are we going in the future?
I don't think small molecules are going anywhere. Basically, they offer extraordinary richness in terms of their tailorability. They offer the benefits of oral dosing. They offer the benefits of straightforward formulation and stability. So I think it's a modality. They're incredibly fruitful and sustaining approach to addressing important diseases. And I think with the improved understanding of signaling networks, presenting opportunities for targets that involve protein-protein interactions, targets that involve cryptic pockets, targets that involve allosteric modulators, it's just going to be a continued space of opportunity for small molecules as a modality.
And projecting out still 2050, where's Iambic today? Or today being 2050?
Absolutely. We're building a company for the long haul. We've been fortunate to recruit a team that has the experience not only to execute where we are, but to execute in the upcoming stages of the clinical progress of the company. And I think that is going to hold us in great stead as we push towards 2050, as you say. And I think that what we're really going to be able to see is expansion to new therapeutic areas, the continued leveraging of the platform to execute across different mechanisms of action and protein target classes, and the ongoing innovation on the front of the high throughput experimentation and the AI. Iambic is a company that was founded on that innovation, and we see that as a essential and sustaining part of who we are as a company.
Oh, absolutely. I mean, Tech bio is, of course, a space where we love to engage, and everything from the physics-based AI incorporation to microfluidics, high throughput biology, chemistry, tying those together, it's clear you've built something powerful. And I want to pivot this in a slight direction, but as someone who has built such an incredible company, do you have any advice with current graduate students or professors who might be interested in making that jump to entrepreneurship themselves from academia and maybe starting a company of their own?
I think that's something you have to think about very carefully for yourself. Making that jump is something it's not for everybody, but it is absolutely for some people. And I would say that for me personally, the opportunity to not only create tools that are scientifically important, but to then have the pleasure of using those tools to make a real world impact, to really see in a strikingly short period of time that translate to human impact is something that is a tremendous opportunity to participate in, and I would consider that among the balance of factors.
No, that's very fair. And I know, as you mentioned, you enjoyed being an academic. So thank you for sharing that dual perspective. There aren't as many folks nowadays who have both, though it's starting to become certainly, of course, more common. And as we talk about this transition to the clinic, and you talked a little bit about hiring the right team, making sure we have the right people involved, is there more you can share from that front? How do you ensure you're doing your best to not only build that interdisciplinary team on the R&D front, but also now at that clinical stage? Who do you think about from a board perspective, an investor perspective, and how does all of that come together to really ensure you're bringing the best chance forward for patients?
I think that's it's a great question, and it's a multifaceted question that you're asking. Yeah, it's true for the internal team, for the company itself, making sure that you have the senior executives that have done it before, that have those decades of experience of actually, navigating the complexities of clinic, and that have seen the transition of programs from preclinical to clinical. So that's of course one element of it. But in terms of surrounding the company with the right advisors, with the right board members, with the right investors, that's another incredibly important part of ensuring that that the company is positioned for success in doing that. Entos has been very fortunate in having investors from the early days that spanned from technology to clinical biotech experience. Even at our Series A, we were fortunate to have Catalio and Co2, Sequoia and OrbiMed, and now we're able to have Ascenta and Abingworth and NVIDIA as investors in this Series B raise, and it really reflects that duality of the company, not only on the technology side but also on the deep clinical side for building a biotech company to succeed. And in terms of advisors, having people who have done it before. Entos has been very fortunate to recruit to our board of directors Bill Rastetter, the CEO of IDAC, and so many other things in the last year, and that's been a huge benefit as we make this transition. We've announced a world-class clinical advisory board, including Alix and Maria Kohler and Kwok-Ken Wong, and we've always benefited from an extraordinarily strong scientific advisory board as well, with founding members that include Francis Arnold and Derek Lowe, and with more recent additions that include Nick Sakamano and that include Tom Daniel. So really making sure that the company is surrounded with the diverse voices that help you to think through the challenging situations and bring that benefit of experience.
I love the way you phrase that around diverse voices. That's a great consideration, especially as you bring together all of these different players. So it sounds like you're more than ready to move forward to the clinic, which I'm going to be thrilled to see. So I know we've talked about a lot of professional things today. Before we wrap up, would love to maybe just ask a quick personal question and say, Tom, what do you like to do for fun? Obviously, you're not building one of the leading small molecule and physics-based AI platform companies. How do you like to spend your time?
I have two young kids, and I do a lot of kind of parent stuff with the kids, and I derive a lot of pleasure from that. My daughter and I play a lot of music together. She plays the violin and I play the cello, and my son has also just started the cello. So we do a lot of that at home, and that's a lot of fun. And I play various sports badly, so it's also good.
Oh, I love that addition, and maybe we can play them badly together sometime. And before we wrap up, any other closing thoughts, shameless plugs, anything you want to share with the audience that maybe we haven't gotten to today?
In terms of shameless plugs, Entos is hiring. We have an amazing team. We've got amazing trajectory, and we're about to embark on an incredibly exciting new chapter of the company. And across all areas, preclinical and clinical and science and technology, we're looking for amazing new people to join us in that journey.
No, it's always it sounds like a great team to be a part of, and always good to hear that company's doing well. So, Tom, thank you for an absolutely fantastic conversation today. Very grateful for your time and sharing Entos, now Iambic story. Thank you again.
Appreciate it, Chris. Thank you.